CH, CH2, OH, 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 this is glycerol and triglyceride. R, R1, R2 and R3, these are fatty acids. Okay, these are fatty acids and esterified with the glycerol. Okay, and then phospholipid, it is an example, and lecithin, phospholipid and lipids that has phosphorus and phosphorylated organic compound. You see there is N, CH3, 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 this group, and R1, R2, these are the fatty acids. Okay, and this is it. Now glycerol and phosphor is present, so four groups are present. And fats and oils, these are lipids. Like cholesterol, it is a steroid kind of thing, and these sterol is also a lipid, kind of lipid. And then adenine, that is purine, that is two heterocyclic ring is present. Then adenosines, that is adenine, and that is sugar is present, so this is adenine. And then adenalic acid, a phosphate group is also present, phosphate group and nucleoside. And uracil, that is pyrimidine, that is nitrogenous base. And then nucleosides, and then nucleotides. Okay, so this is the small molecular weight organic compounds. Now primary and secondary metabolites. Now primary metabolites, these are the metabolites that involved in the physiological functions of the living organisms, normal physiological functions. Okay, these can be amino acids and sugars. And in secondary metabolites, and they are the so through the metabolism of the primary, okay, through the metabolism there are some, no, just remember the secondary metabolites, these are without knowing any normal function in physiological processes. So these are secondary metabolites. Example: alkaloids, flavonoids, rubber, essential oil, antibiotics, color pigments, scents, gums and spices. And they do not have normal functions. Okay, so these are secondary metabolites, and they are useful to human welfare. They are rubber, drugs, spices, scents and pigments, and they have ecological importance. Now look in the diagram, that is some secondary metabolites like pigments, carotenoids, anthocyanins, alkaloids, morphine, codeine, terpenes, diterpenes, monoterpenes, essential oils. These are lemongrass oil. Okay, toxins, urbine and ricin, lectins, concanavalin, drugs, vinblastine, curcumin, and polymeric substances, rubber, gums and cellulose. So these are the secondary metabolites. Remember, essential oil, lemongrass oil, right? So this is it. So these are the secondary metabolites. Okay, and then there is uh, like some biomacromolecules. Now we study the biomicromolecules. Now biomacromolecules, like biomicromolecules, they have a molecular weight 18 to 800 dalton, and but biomacromolecules more than 10,000 daltons. Okay, more than 10,000 daltons, 1000 daltons. Okay, less than 1000 dalton micromolecules, and more than 10,000 daltons, they are 10,000 daltons and above, they are biomacromolecules. Now these are acid insoluble fractions, has four types of organic compounds, that is proteins, nucleic acids, polysaccharides and lipids. And biomacromolecules, like amino acids and sugars, and all these are like carbohydrates. No, not carbohydrates or sugars, like monosaccharide type of things. They are biomicromolecules. Now exception is the lipid. As I told you, molecular weight do not exceed 800 dalton, but they are still the lipids are not strictly biomolecules, and they form the vesicles and get separated along with the acid insoluble food. Okay, now acid soluble food represents the cytoplasmic composition, and macromolecules from cytoplasm and organelles form acid insoluble fraction. Now everything is covered. Remember, carbohydrates is 3% of total cellular mass, and lipids is 3%, lipids is 2%, ions is 1%. Now proteins, they are polypeptides. They are amino acids linked by peptide bond. These are proteins. They are linear chains. There are 20 types of amino acids: alanine, cysteine, proline. Proline is an amino acid. Okay, now they are now protein, they are heteropolymers, right? They are not homopolymers. Okay, and then there are like some dietary proteins, and they are essential, non-essential amino acids. Essential, that means our body do not synthesize it, and is non-essential, our body can make it There are 8 non-essential amino acids that we needed. Phenylalanine, leucine, isoleucine, like that, given in ncert chemistry or biomolecules. Go and watch it. And then now proteins carry out many functions in living organisms. Now some transport nutrients across the cell membrane. Now some fighting infectious organisms. Some are hormones, some are enzymes, etc. Now collagen is a protein. It's most abundant protein in animal world. This is ribulose bisphosphate, carboxylase, oxygen. Now rubisco is the most abundant protein in the whole of the biosphere, especially in the plant, right? So this is it. Now some proteins and their functions. So collagen is intercellular ground substance. Now trypsin is an enzyme, insulin is an hormone, right? So antibodies fights infectious agents, right? Now receptor, there is sensory reception, that is smell, taste, hormone, etc. That is that is enables glucose transport into the cells. So this is it. Now polysaccharides, now found in acid insoluble pellet and polysaccharides. These are carbohydrates. These are long chains of sugars. Okay, and sugars, just like carbohydrates, and sugars and polysaccharides both the same name. And then cellulose. It is a structural polysaccharide and made up of glucose, and cellulose is a homopolymer. And now the starch and glycogen. Starch and glycogen both are storage bodies found in plant and animals respectively. And inulin is a polymer of fructose. Remember fructose. And glycogen has branches as well, right? And now in starch they found the helical secondary structure. This is why they hold I2 and they show the blue color. And cellulose does not have complexes, so they do not hold iodine, so they do not give blue color. So this is it. You see in the diagram there is glycogen and you see OH. Two glucose molecules are linked. They are dehydrated at like 1 2 3 4 4 1 4 1 position, you see in the diagram. And this is it. And now going to like plants, cellulose is made for the paper. We made it paper like cotton fiber. Okay. And more complex polysaccharides in nature. Now amino sugars, and chemically they have amino sugars like amino group and sugars. So amino sugars and chemically modified sugars. Example, glucosamine and N-acetyl galactosamine. So glucosamine and acetyl galactosamine, right? Now exoskeleton or arthropods, they have kinetin. It is complex polysaccharide, and these complex they are mostly homopolymers. So chitin can be homopolymer, just like homopolymer, right? Made can be made like glucosamine and N-acetyl galactosamine. Now nucleic acids, RNA and DNA. These are nucleic acids, polysaccharides and polypeptides. So these are true macromolecules, right? And for nucleic acids the building block is a nucleotide, right? A nucleotide has three chemical distinct components. One is heterocyclic compound, second one is monosaccharide, and third is a phosphoric acid or phosphate. So phosphoric acid or phosphate, right? Now you see adenine, guanine, uracil, cytosine, thymine. Adenine, guanine, these are substituted purines, while rest are substituted pyrimidines. Okay. Now okay, the sugar found in polynucleotides is either, now polynucleotides, these are nucleic acids, now can be either ribose, monosaccharide, pentose, or 2-deoxyribose. Now nucleic acid containing deoxyribose is deoxyribonucleic acid, that is DNA. And which contain the nucleic acid, that is polynucleotides, contains the ribose, is called ribonucleic acid. RNA. Now structure of proteins. Okay, they have primary structure or they have secondary structure. Now the proteins, so proteins have primary structure, secondary structure, tertiary structure, and quaternary structure. Quaternary structure example is hemoglobin, that has two similar subunits, that is two subunits of alpha type and two subunits of beta type. Okay, there are four subunits, but two of these are identical to each other, and more than one polypeptide or subunits, they form like architecture structure, that is quaternary structure, and intertiary structure, there is hydrogen bond and disulfide bond, and hollow wooden like structure they form. They have the active sites which is required for biological activities of proteins. And okay, now in secondary structure there are alpha helix and then beta plated sheet, you see in the diagram, like, okay. And in proteins only right handed helices are observed, so right handed helix are observed in the proteins. Okay. And the in the primary structure you see there is first, so it just the long chain of polypeptide, the linear chain. They have the first amino acid, that is the N terminal. They have last amino acid, that is C terminal. So this is it. You see the first, that is left end, that is I think left end, that is non-reducing, and right end, that is reducing it. So this is it. And then there is like enzymes. Now what about the enzymes?